welcome to issue 34. I'm here with Natalia from Sam Houston State University. How are you?
I'm good. How are you?
I'm good. So you go to Sam Houston State University. What do you do there?
Yes, I am a doctoral student in the forensic science program.
Nice. What year?
6.
That's fine. Nice.
A lot of research to be done, so it's taking me a little bit longer.
But that's okay, because good research, I hope.
Yes, very good. We got a lot of good collaborations happening at Sam Houston.
Okay. And what brings you to issue 34?
I'm actually presenting one of our collaborations. I have a poster presentation. We partnered with QIAGEN to do the project, and we wanted to examine whether we could determine a decision point where we have degraded samples, and we decide, do we go the traditional STR CE-based chemistry route, or do we do NGS?
Okay.
And see if we can pick up more information that way.
Okay. And so this gets implemented before any testing has been done with that sample? Or where in the forensic workflow does this get implemented?
So we're thinking that the quantification step could be where the decision point lies. We examined both the quantity of DNA in the sample and also the degradation to see if either one of those could serve as the decision point.
Okay. And so tell me more about how this decision would be made.
So we looked at the allele recovery at the downstream part of the workflow and compared it back to the quantification information to see if any samples that didn't do so well with traditional STR typing on the CE did or had improved results using the Mainstay kit.
Okay. So at the quantification stage, an analyst will analyze the degradation, or I guess DI information along with the quantity, and then at that point they would themselves make a decision on whether it should go the traditional route versus a different route?
Yes, so what we found was actually the degradation index was not as informative as we had predicted. Okay. It wasn't a consistent result when we compared our DIs to our allele recovery, but we did sort of see a pattern with examining the quantity of DNA and then comparing that back to our downstream genotyping.
Okay, nice. So during your tenure at Sam Houston State University, how has it been?
It's been great. We have a lot of good collaborations. I really enjoy the professors. We have so much hands-on experience at SAM that I couldn't think of a better place to be a doctoral student.
Yeah. Is this your first time at ISHI?
Yes, actually.
How has it been so far?
It's been great. I really enjoyed a lot of the speakers. I'm excited for a lot of the FIG coverage. That's another interest of mine that I'm slowly building up. And so I had a table talk lunch over that today and a lot of great presenters, I think, coming tomorrow.
Nice. So what's the one thing that you have been looking forward to most outside of FIG?
Outside of FIG? I think just learning more about NGS. Okay. I think that's where everything in DNA analysis is heading towards. And so being able to do a project that includes using Mainstay. I have another project I'm working on that uses Kintelligence. Okay. I'm soaking all of this great information up while I can.
Yeah. So I'm not as familiar with Mainstay. What is it?
So Mainstay is QIAGEN's NGS kit that includes not only autosomal markers, but YSTR markers. Okay. And so that's why we wanted to compare it back to the standard STR kit, 24-plex, to see if there was any advantage in having those extra Y markers, if we could pick up that information in degraded samples. And it turns out we can. We're getting more genetic information by using Mainstay, so it might be the way to go in the future.
Nice, nice. So post-graduation, where do you see yourself going? Or what would you like to do?
Ooh. It's funny that you ask because I do all this DNA work, but I think my passion lies in CSI. And so I want to find something that can blend the two and then maybe perhaps even incorporate FIG.
Nice.
So I don't know if that exists out there, but I'm going to search long and hard.
You can make it happen.
Yes.
Nice. Is there anything else you would like us to know about your research or about your poster presentation or about you?
Yes. So with the research, the poster just includes a smaller number of samples, but we are going to expand that research and not only examine different types of samples using Mainstay compared back to 24-plex, but we really want to try and expand using SNP typing. So the Forensic Signature Prep Kit, Kintelligence, to see if we can add in those types of kits in our decision point assay.
Okay. What sample types have you analyzed, and what sample types do you want to expand to?
So right now we started with 2 femurs from 2 different cadavers, and it's really interesting how we got our samples. We actually thermally degraded them. So we burned the bones at different times, or for different lengths of time, I should say, to get different colors, so like different burn points.
Okay.
So we obviously have our unburned control, and then we have our light brown, brown, and black. And so increasing levels of degradation we're seeing with those. We want to expand and not only do burned bone, but other types of damaged bone, buried, waterlogged, things like that, and just look at other degraded samples as well.
Nice. That's very interesting.
We have a great willed donor facility at Sam Houston, the Southeast Texas Applied Forensic Science Center, where people can donate their bodies for scientific research. And so we're very fortunate that we have these samples that we can utilize in our projects.
Yeah. It's kind of like you're working with actual real samples, how it would be applied.
Exactly. Exactly.
Nice. I like that. Well, thank you so much for speaking with me.
Yes, thank you for having me.
Enjoy the rest of ISHI.
Thank you.